This is lecture 8, the first part out of 3, and we'll start with chapter 22, microbial disease of the respiratory system. As you know, the respiratory system exchanges gases between the atmosphere and the blood. We have the upper and the lower respiratory system. I am not going to go into the details, but please know your components of the upper and the lower, since we're going to be discussing the infectious disease that actually affect these systems. These are the structures. And we're going to begin. We do have normal microbiota in the lower and upper respiratory system. They are typically not present in the lower respiratory system. However, in the upper, we do have normal microbiota, and they actually help us to limit growth of pathogens. However, they can also become opportunistic pathogens. And here we have the normal flora of the respiratory tract. Tell me why. Okay, so here are the common bacterial virulence. These are your bacteria. And as you can see, in the upper respiratory, you have the common cold, dentocaries, pharyngitis, otitis media. And then in the lower, you have the croup, whooping cough, bronchitis, and bronchiolitis. So we're going to start with the streptococcus respiratory disease. One of them causes pharyngitis, which is your sore throat. And it is often accompanied with fever, malaise, and headache. Laryngitis and bronchitis can occur if this spread to the lower respiratory tract. It can even get to rheumatoid fever or scarlet. And even so, it can even actually go to your kidneys and give you glomerulonephritis. And when the doctors examines the patient, they look for the pus pockets in the inner mouth, in the throat. Okay, so what are the signs and symptoms? Well, it's caused by group A streptococci. This is a gram-positive bacteria. And basically, your M proteins, they inhibit the complement 3B. And you also release hyaluronic acid. And this actually avoids phagocytosis from your white blood cells. And another way to actually see if you have this virulent factor, they look at two enzymes such as C5ase, peptidases. We look into the pyrogenic toxins, which are the ones that actually stimulate the fever. And other toxins, which are also known as erythrogenic. And the streptolysis is the one that actually lyses your red blood cells, your white blood cells, and your platelets. It actually occurs when your microbiota is not as abundant, meaning it's depleted, and there's a large inoculum, and then there's adaptive immunity that is impaired. It spreads through your respiratory droplets in the air, and is more often in the winter and spring, which is not our case locally. The diagnosis, sometimes they think it's viral, but it's not. What treats it the best is oral penicillin. Now, if you look at all these ailments, they all have very similar symptoms. Therefore, that's why it's important to do what? What are the lab results going to give you? Diphtheria. Diphtheria gives you sore throat, fever, and it is when you analyze the actual patient, you can see the pseudomembrane that actually obstructs airways. This is dead tissue that is caused by the diphtheria toxins. The lymph node of this patient also swollen, and it gives them the appearance of bull neck, which is what you see right here. It is caused by a bacteria known as Corynebacterium. It looks like a corona if you look at it under the actual scanning microscope. The vectors that are virulent, it produces toxins, and then it also prevents the polypeptide synthesis and therefore inhibits the translation elongation factor of your normal proteins. It is a gram stain of the Corynebacterium, and as you can see, it's a gram positive. Another way to actually do a diagnosis, they look at the antitoxin strict samples. When they treat with these antitoxin antibiotics, it actually gives these presence in the plates, in agar plates. This used to be the leading cause of death among immunized children. Bacterial disease of the upper respiratory system, rhinosinusitis, and otitis. What are the signs? Well, again, malaise, headache, and you have your nasal passage to be actually very inflamed. The otitis media, which is in your ear, has severe pain. The pathogenesis and the epidemiology. The bacteria in the pharynx spreads to your sinuses via the throat. The rhinosinusitis is more in adults, but the otitis is more in the children. Saline solution helps us to actually get rid of these symptoms. But watch out. Why is that? Tell me why. Okay, now we're going to go into the viral disease. Well, your common cold. What's the difference between the common cold and other viral disease? Well, one of the indications you have a common cold is that you don't have fever. Okay, the other symptoms are present, but there's no fever. The pathogens are caused usually by your enteroviruses. There's many viruses that can actually cause the cold. These rhinoviruses are very good at infecting other through droplets, between patients. How do you get these droplets, respiratory droplets to spread? Well, coughing, sneezing, even through objects that you contact with a person to person. The diagnosis, usually antiviral. Okay, you actually know it's virus because you have a pliconaviral, which can reduce the duration of the symptoms. But the best way to actually prevent it is through hand washing. This shows you the rhinovirus capsid, which is a protein. Your piconovirus, how does it actually attach? Well, it has receptor binding that actually allows the entry into your cell. And as you know, viruses, what do? They use the transcription translation machinery of the host in order to replicate. And once they have actually produced the capsid, they egress, meaning they can do cell lysis of the host. Tell me why. Why not use penicillin or erythromycin or ciprofloxacin? Okay, so now we're going to go into the lower respiratory organs, which are usually axenic. And that pretty means that they don't have any bacteria whatsoever present. That's what axenic means. The infections of the lower respiratory system They can lead to life-threatening illness, and you are familiar with them, which are bronchitis, bronchiolitis, and pneumonia. And here are, as you can see, CRAP, whooping cough, bronchitis, and bronchiolitis. Okay, this is your bacterial pneumonia, and how is it detected? Well, X-ray. As you can see right here, there's a lung inflammation, and your alveoli get fluid filled. The bronchioles also. You could have the affected region, and it causes the disease known as your lobar pneumonia, also your mycoplasma pneumonia, which is also known as your walking pneumonia, and you also have your healthcare associated pneumonia. The most serious are your bacterial pneumonia, and that refers in reference to your viral pneumonia versus the mycos pneumonia. Okay, the pneumococcal pneumonia, well, your first sign again, fever, chills, chest pain for sure. You have a shortness of breath and then rapid breathing at the same time. And the problem is that your blood enters the lungs and then it causes the rose-colored sputum, which is what you saw in the X-ray. Streptococcus pneumonia is the cause, the bacterial, and we look for virulent factors, which are the adhesins. These are proteins allow the streptococcus to adhere to the host and then get into the host. They also have a capsule, and as you know, capsules can actually work very well against your phagocytosis from the white blood cells. And your neurolysins is a neurolysin pore is actually a protein that causes the cell lysis, and therefore it damages the tissue of the lungs. And here is the capsule, and again, this is a transmission electron microscope slide. You cannot see this under the normal microscope, or rather the light microscope. This pneumococcal pneumonia, okay, you inhale the bacteria through respiration, of course, and the host actually destroys the immunoglobulin A protease. Is the one that destroys your immunoglobulin. That's the enzyme. How do we treat it? The best treatment here is penicillin. However, it can also prevent it with vaccination. First sign again, same thing. All of the pneumonia has the first signs of your fever, malaise, sore throat. However, you have excessive sweating and a non-productive cough. That means you cough, but nothing comes out. This is your atypical pneumonia, which is also known as your walking pneumonia. Basically, you might have this for weeks and you're coughing and coughing and coughing, but you don't even know you have it. But it can go into great danger. Okay, your actual mycoplasma pneumonia is the most common in teenagers and the young adults. And since you have it on for such a long time, it is actually difficult to know you have it. That's why they say it's difficult to diagnose. We treat it with a typical unresponsive beta-lactams treatment. That means that we have to use erythromycin or doxycycline. The prevention is difficult because most of the time when you have a walking pneumonia, you don't even know you have it. It's asymptomatic. Let's move on to Klebsiella pneumonia. These ones are the ones that give you a thick bloody sputum. This is the pneumonia that you probably heard through history when a patient coughing actually have a bloody sputum. They have a huge capsule. The Klebsiella pneumonia has a huge capsule, so that's what we know it is Klebsiella. The people that have a low immune system are the ones that are greater risk, and we actually have to treat it with antimicrobial drugs. And the way to prevent it is good aseptic techniques. This is actually a leading cause of nosocomial infection. What is nosocomial infections? Think about that. And these are the capsules that you can actually see with the capsule staining. As you recall in lab, you probably did a negative stain. And then you see the empty spaces, which are your capsules. Other pneumonias, some of them are done by Haemophilus influenza, even Staph aureus. Yersinia pestis, this is the one that actually caused the pneumonic plague. You have the chlamydophila. This is the one that is actually caused by birds to humans, a zoonotic. And your chlamydophila pneumonia. Legionnaire disease. It is called this way because it happened to occur in a veterans convention, which were the Legion, and it was actually spread through all of these people that were in the convention. Well, not all of them, but the ones that were actually at a higher exposure and older age. It is called by Legionella pneumonophila. And he actually kills the human cells. He may cause tissue damage and inflammation into your lungs. We can actually identify it because it's activated in charcoal. This is a charcoal yeast extra agar. And when you see here, right here, it becomes even fluorescent in the media, so you could tell this is the Legionnaire. Okay. And it loves to live in moist areas such as your air conditioners. And again, like I told you, it was the first outbreak in the Legionnaires Convention in the early 1970s. How do we treat it? With antibiotics, such as the quinolones or the macrolides. TB, also known as tuberculosis. It is still the leading disease killer in the world. It has declined, but it still is there. Minor cough and mild fever is the first sign. However, your symptoms are not always there. It's caused by mycobacterium, so that means it is identifiable through acid-fast stain. It has defense mechanisms that it protects from phagocytic lysis from your white blood cells. And it's hard to kill because it's resistant to many antimicrobial drugs. When you do an acid fast, you can actually see these TB cores, like you see in this area right here. So they actually have adhesins proteins that allows them to attach to the white blood cells. It is airborne. You have three types: primary, secondary, and the third one is disseminated. That is as they progress through your lower respiratory system, which is here. As you can see in the beginning, it gets to your alveoli, and once it gets into your alveoli, it's going to attack your white blood cells. It's going to attack them right there, and it's going to go in them. And it stays there, and it causes the actually what you call caseous necrosis, which is death of your cell, of your white blood cell. And then once it has replicated enough, it's like boom, it explodes that cell, and it gets out of your alveoli and attacks the other ones. Tuberculosis, it affects 1/3 of the world's population. That is right. However, most deaths are actually in Asia and Africa. How do you know if you detected tuberculin skin test? However, the problem is that if you were given a TB vaccine, especially in the early 80s and before that, when you take that test, it gives you a positive, but you don't actually have it because it detects the tuberculin protein. The tubercles actually are seen in the x-rays. We have new drugs. However, tuberculosis have emerged to be resistant to these drugs. The Bacillus calmette-guerin BCG vaccine is actually available, and that's the one that is used against tuberculosis This is an example of the diagnosis of the skin and lung. And this is the one I was telling you that I showed you for a positive, but it could be a false positive. The whooping cough, also known as pertussis. It looks like you have a cold, but then the cough develops. It is caused by Bordetella pertussis, and it has numerous factors as far as virulent, and some of them includes the adhesin, which is the protein that allows for these actual bacteria to adhere to the membrane of the host and then get in. Okay, we look at to pathogenesis. What does it do? It incubates, then it causes inflammation, and then it goes into the symptoms, and then you have a recovery. However, then it reattacks the patient. It is very contagious. It is airborne. However, the bacteria doesn't survive a long time outside the body. Your diagnosis, again, is by what we actually just discussed, by the whooping cough, and it is prevented with a vaccine known as DTaP. Let's move on to anthrax. It looks like you have a flu, but it's not. Then it moves on to a very severe cough, lethargy, and you go into shock and even death if not treated immediately. Okay, the Bacillus anthracis is the one that causes it, and they have a capsule and actually toxins that actually are the variance factors that are to be identified. It is not spread from person to person. However, it is acquired by contact or inhalation of these endospores. As you know, the bacillus can do endospores when put into critical environment. They look into the bacteria in the sputum, and then the must treat it immediately with aggressive treatment. That means if they don't do it, it will spread very quickly throughout your lung systems and respiratory tracts. It is given to selective individuals, and those individuals the vaccines are given are, again, people that are in the services of the government. Tell me why. The flu. The pandemic of the flu, known as the Spanish influenza, started in the 1918, and it actually didn't go to 1919. It actually went on to 1923-24, and it actually killed more people through Europe than World War One, more than 20 million. It is caused by the virus known as influenza, and it can actually lead to pharyngitis. And of course, we know that we have many types of flu. It has gone into mutations, and the new strains are like H1N1, H1N2, and so on and so forth. It actually occurs with antigenic drift or antigenic drift. So what does that mean? Well, as you can see here, your influenza virus enters, okay? And that is your antigenic drift. It mutates within the host, and then it comes out as another influenza virus. That is known as antigenic drift. However, in the antigenic shift, you have two viruses that enter, and then they release the actual genetic material and emerges together, and then as it comes out, you have another influenza virus as one that were coming from virus 1 and 2, and they become 3, and that's why it is antigenic shift. Okay, so how do you know you have the flu? Well, of course, you have a virus that enters your body and it causes the fever, a lot of shaking, and then if you are not to be treated immediately, you can be susceptible to secondary infections. Okay, and this virus can, of course, cause a lot of damage to your epithelium in the lungs. How do you diagnose it? Well, It depends. It depends on the type of virus and the time that it occurs. Um, some of this flu comes early in the winter, some come late in the winter. It depends on the community outbreak where it is diagnosed. Okay. The treatment, some of the infections can be actually uh, treated to inhibit the neuraminidase, which is a natural enzyme that is antigenic determinant, and you can be prevented by vaccination. So this is your coronavirus, and as you can see in the picture, they call it coronavirus because it looks like a corona, like a throne. Okay, uh, these are two emerging diseases that can be either severe acute respiratory syndrome or the Middle East. High fever, shortness of breath, again, dry cough and pneumonia. They can lead to pneumonia, and then they're most usually mild, but they can actually get into SARS and MERS, which have very much higher fatalities. And this is the Middle Eastern one, of course. How does it spread? Well, it goes from the lungs, but it can actually go all the way to your heart and kidneys. It is confirmed that you have it by isolating the virus, and then they actually do antibody against the virus. That is basically they look at the antibodies antigens connection, and the treatment happens to be supportive. Okay, the respiratory syncytial multinucleus virus infection. These are known as your RSV virus, and they actually also target your lower respiratory system, and it looks like a cold-like symptom in older children and adults. However, it can actually damage your lungs very much if it's not treated immediately. How do they actually get in to your cells? Well, they form like a hairpin structure. As you can see here, you have two proteins that are right here, and it goes and connects between the two membranes, the viral membrane and the host membrane. And once they are connected together, they fuse together and the virus gets in. As you can see, it's right here. This is now a cell that is infected. But once the cells that is infected, they connect the two cells together like the hairpin I was mentioning earlier, which allows the channel to be formed in which the cell is infected from the actual RSV virus. Okay. It is diagnosed by immunoassay, and that is most of the time with an ELISA assay. And the accepted techniques is the best way to avoid it. Hantavirus. Hantavirus is the one that causes the hantapulmonary syndrome, and it's actually transmitted from mice via inhalation. It goes to your body via blood first, and then it goes into your respiratory system, leading to a shock. And as you can see here, the hantavirus is made up of 3 negative single-stranded RNA molecules within the capsid. Okay, the way to transmit it is person to person, but it doesn't occur. It actually comes from mice. There's no treatment available, and if you want to prevent it, get rid of the rodents. CMV virus and the metapneumovirus, these are the second most common cause of the viral respiratory disease, okay? And then you have the parainfluenza virus. It has 3 strains, and they can actually are the ones that cause CRAP. And CRAP, of course, is, well, not occurs, but it actually occurs only in young children. Tell me why. Okay, so now we're going to go into the fungal disease. And as you can see, we have the coccidioidomycosis, blastomycosis, and the histoplasmosis. Okay, the coccidioidomycosis. The coccidioidomycosis, it looks like you have pneumonia or tuberculosis. It can become systemic, and you know what that means. That actually can go throughout your entire body systems, but it's more in the immunocompromised people. Okay, they assume like a yeast form at the human body temperature. Okay, that means they're facultative, and they actually come from the soil, and they actually go into the body through inhalation as spores. Okay, as you saw the maps earlier, this is most likely in southwest US and northern Mexico. And it is treated with amphotericin B. How do you prevent it? Wearing a mask. Okay, another mycosis of the lower respiratory system is blastomycosis. It looks like the flu, or it feels like the flu rather. It could be systemic, and it's caused by the fungus known as blastomyces dermatitis because it actually affects the skin. As you can see right here. How does it enter your body? Through inhalation, okay, with the dust. And as you inhale the dust, you actually have the spores from the fungus. And it is diagnosed again by looking at the fungus in clinical samples. It is also treated with amphotericin B, and it is known to relapse in AIDS patients. The third is histoplasmosis. It is asymptomatic most of the time. However, if you have symptoms, it can cause blood in your sputum when you cough, and even skin lesions. It is caused by Histoplasma capsulatum. It looks like it is a pathogenic yeast, okay, at human body temperature. And this fungus actually avoids macrophages from white blood cells. It is actually more prevalent in the eastern US. And how do we know which one it is? Again, identifying the fungus in clinical samples, which is actually now used with molecular biology. It is more prevalent in the immunocompetent individuals. Which means that if you have good health, you don't need much treatment, you'll be fine. PCP, also a type of pneumonia, pneumocystis pneumonia. Just like other pneumonias, you have difficulty breathing, anemia, low oxygen levels in the lungs, and you also will eventually cause a fever. Caused by pneumocystis jirovecii. Okay, it is again in the droplets in the air. It is very common in AIDS patients. You again look at the clinical and microscopic findings to find out which one of the fungus infection you have. Treated with trimethoprim and the sulfamethoxazole, which is a drug. The only problem with this drug, they can actually also affect your liver in the long run. And to prevent it is actually impossible. If the spores are present, there's presence. Tell me why. And this concludes lecture 1 of 3 that are all of lecture 8.